Hello everyone, it is my great honor to present to you the research on sex-specific associations of Roseburia with uric acid metabolism and hyperuricemia risk in females. Hyperuricemia affects approximately 14% of the population, with a marked gender disparity showing lower incidence in females. While the role of gut microbiota in HUA has been extensively studied in males, its influence in females remains unclear. This study recruited 623 participants, including 270 females, to explore this gap using 16S rDNA sequencing. These findings shed light on gender differences in HUA incidence from a gut microbiota perspective and propose Roseburia as a promising target for probiotic-based prevention of HUA in females. Highlights. First, analysis of 623 population samples suggests microbiological factors behind lower HUA incidence in females. Second, Roseburia is a sex-specific genus that may benefit the mitigation of HUA in females. Third, Roseburia is positively correlated with E2 levels and negatively correlated with UA levels. Fourth, the xanthine-glycine pathway is positively correlated with the abundance of Roseburia, which might contribute to lower UA levels through this pathway. The results of the clinical indicators show that levels of urea, creatinine, cystatin C, and uric acid were significantly higher in male_health than in female_health. The subject's gut microbiota and clinical indicators were comprehensively analyzed. The Mantle test results and Adonis test suggested that gut microbiota may influence these clinical indicators and potentially serve as indicative markers of HUA. Further quantification using Bray-Curtis distance revealed a more pronounced difference in gut microbiota structure between healthy males and males with HUA. Based on the established significant association between gut microbiota and HUA, the relationship between gut microbiota and sex differences in HUA was explored. The gut microbiota genera screened from healthy males and females may represent those with potential protective significance for females without disease. The results indicated that bacterial genera capable of producing short-chain fatty acids were enriched in the healthy female cohort. To further validate our research, 256 data on gut microbiomes from 3 published studies on HUA populations were collected to examine the abundance differences of these genera across different populations. The negative correlation between Roseburia and UA levels was significant. Subsequently, a model of the random forest was applied to assess the predictive importance of these 14 genera, with Roseburia emerging as the most important in distinguishing between female_health and female_HUA. Therefore, Roseburia may serve as a key sex-specific protective genus that helps females resist HUA. Roseburia had been identified as the marker genus that most effectively distinguished between premenopausal and postmenopausal females. Spearman's rank correlation analysis showed that Roseburia significantly correlated with E2. E2 levels were significantly higher in the female_health group than in the other 3 groups, consistent with the abundance profile of Roseburia in the gut. Lactate dehydrogenase and hypoxanthine phosphate acyltransferase, associated with UA excretion and metabolism, were more abundant in the female_health group than in the female_HUA group. Based on the above results, a potential mechanism was proposed. E2 may promote the enrichment of Roseburia by affecting the gut microbiota, and high levels of Roseburia may reduce the production of UA by enhancing the xanthine-generating glycine metabolic pathway. This study found that for HUA, there is a significant difference between male and female gut microbes and that females have more beneficial genera of gut microbes, which may be one of the reasons why females are less prone to HUA. In exploring the gender-specific mitigating effects of gut microbes on HUA in females, we identified Roseburia as a key genus. Higher E2 levels positively correlated with Roseburia, allowing this bacterium to exert a more pronounced effect on reducing UA levels. In contrast, males lacked this E2-mediated advantage, rendering them more susceptible to HUA. These findings not only enhanced our understanding sex hormones influence human health by modulating the gut microbiota but also provided a theoretical foundation for developing novel therapeutic strategies for HUA.